My name is Lauren Lopez, and I am currently an undergraduate student at San Jose State University. During the summer, I worked along with Dr. Randi Pullen at DEVCOM ARL-South. And I am presenting to you what we have developed. We developed a tool called TULIP, which is short for Tunable Ligand Inducible Plasmid. We developed this tool in order to combat the limitations that industrial biomanufacturing has, because traditional plasmids are constricted due to their limitation on how many plasmid copy numbers that they have, which is dictated by the origin of replication. And that's not very ideal for industrialization. With industrialization, you want to have control over your product. You want it to be reliable, and you want it to be fast, while making sure the yield is high. Thus, we developed TULIP. TULIP allows for us to control these engineered constructs that we have by controlling the amount of DNA we have, as well as how much genetic expression we want, such as if it's too much or too little, we could desire that to whatever we want it to be. So I developed TULIP, and these are the steps that I went throughout making it. TULIP consists of 8 different parts, all being found within a traditional plasmid, just engineered to make sure things are very, very reliable, as well as producible and scalable. So this implies that we have an origin of replication in order to make sure that our plasmid is able to reproduce itself and produce itself over and over and over again. We have a couple parts that are just there to make sure that, hey, I'm doing the job that I'm supposed to be doing, and I am stable. So they're there for more stability purposes. We also have a bacterial portion, which is antibacterial. And we specifically chose SPEC and CANA for antibacterial properties. However, it could be substituted with any other antibacterial. And for a regulatory part, we specifically chose Rep-A and CYM. The specific part of the plasmid is what our inducer is latching onto, thus the ligand part. So with this inducer, it's able to attach onto that part, and depending on the concentration of that said inducer, shows how much growth you're gonna have, the amount of PCN you're gonna have, as well as the genetic expression. So once we had all of these 8 different parts domesticated, we transformed them into mainly 2 different E. coli strands. We had BL21 and beta-10. They're just generic, usually used in the laboratory, in a microbial laboratory. And we picked colonies that we're looking for. They look positive. They're going on the plates just fine. And then we sequence validate it. We validate our sequences to make sure that the constructs that we are seeing are due to us engineering them and not due to mutations that might have happened along the way, since that is a possibility. So once all of these constructs are validated and approved and they get the A-OK, we stitch them all together in order to make TULIP, or model structure. Once we have TULIP built and we see the positive colonies, that is when we perform qPCR, as well as the fluorescence assay. What we use these 2 assays to do is to quantify how much DNA was produced, as well as the fluorescence assay to see how the fluorescence of those different constructs among different varying concentrations of that said inducer. And what was predicted to be found with the qPCR is that there was an increase in plasmid copy number as the concentration of the inducer increases. As for the fluorescence assay, we predicted that the fluorescence would increase as the inducer increases as well. So what does that mean, particularly when it comes to application courses and application when it comes to this said construct? This could be applied to scientists as a module structure that they could drop out the reporter gene and input a desired targeted gene that they would like. So this saves a lot of time and money for those scientists when it comes to developing these types of circuits. And what TULIP can initially be applied to specifically for bioengineering and industrial cases, which is the main purpose of it, it can make field-ready applications of materials to access to the Army, as well as it could advance the Army's capabilities when it comes to biomanufacturing by reducing our dependency on foreign nations when it comes to imported products, which may be delayed sometimes due to maybe geopolitical issues or perhaps there's conflicts, but it allows to make sure and strengthens our supply chain as well as our field and productive readiness when it comes to the Army.